Welcome. This time round on the Stranger Tractor, we sat down with David from Utilium. So David is one of our impact members here at CoLabs, and he's been working on a bio-based and bio-inspired technology that is aiming to replicate the way in which crustaceans and shellfish manage to maintain an immaculate outer casing, and they don't get gunked up like we do with our boats and other man-made things like pipes and all this other sort of stuff where you get a build-up of biofilms. So the technology is a biofilm buster, and it has applications across industry, in healthcare, like honestly anything where you have the potential for a biofilm buildup. This has potential applications. So it's, for us, we saw it as a really exciting and outsized potential for positive impact. Hence why we provided the free space for David to be able to validate and prototype the technology further whilst he tries to raise some funding to be able to bring it to life. We hope you enjoy this conversation. David's a really wonderful thinker and a fascinating human and a delightful member of the CoLabs family. So please enjoy this conversation. David, welcome.
Thank you.
Thank you for coming on the podcast. I know it's been a while. You've been very intentional with us about you'll let me know when the right time is. You'll let me know when the right time is. What do you think made now feel like the right time for you?
Project-wise, I understand what I'm doing, how I'm doing it, and I know I've discovered something extraordinary. So therefore, that gives me a lot of confidence that I'm not just mucking around in the lab. I'm deliberately doing something that's going to be very effective. And I need that. I'm a confidence person, so I need that confidence. Whereas if I talked earlier, I would have gone, oh, well, I'm trying this, I'm trying that. But because I know I have something and I'm on the way to identifying it a lot more, I have a lot of purpose. I need that purpose before I talk.
That makes sense. Yeah, I guess the way that I interpret you is you are a very intentional person. Whether that's intentionally using daydreaming to try and think about your project or even perhaps unintentionally. But there does feel like there is a lot of intention through who you are and what you do. And I'm wondering whether or not that's had, I guess, a lot of an impact on your career that you've spent, because before you were in the startup land, was it 2-3 decades in sort of biomed, biotech? How would you define
Just medical research.
Yeah. Cool. And I'm curious, so you're not everyone has made as interesting a discoveries as you have, right? Did you want to talk us through some of your research, maybe your PhD?
So I was in research. So I worked as a research assistant for quite a number of years when I finished my undergraduate, and eventually the guy I was working with said, you should do a PhD. I said, what projects you got? He says, well, I got one on AIDS, because AIDS was just starting at that time, trying to work out the virus and everything. And another one, I've got this enzyme that controls energy metabolism. Others have been working on it around the world for 10 years, but I think you'd be quite good at it. So I chose that one. It was more interesting than the viral side of things. And all it was, in those days, there was no internet. You had to wait for papers to come out, go to the library and photocopy them and read them, and no AI. Everything was very old-fashioned, and compared to now, so had to do other abattoirs and things you're probably not allowed to do now. And after a year or so, I was following all their protocols and wasn't really getting anywhere. And then one day I was gardening, I remember, in Montana, where I lived, and I had this idea on a weekend.
Bring the mic a little bit closer. Or that's perfect.
To use like a Velcro, create a Velcro, work out how this activity stuck to a surface it liked. We had some data on that. Called the boss, supervisor, he said, that's good, let's get the team together. We did, we organised a strategy, and a month later, we'd isolated this enzyme called AMP activated protein kinase. And what that means is a kinase is an enzyme that transfers a phosphate from ATP onto a other protein. It changes what it does. And the AMP is a nucleotide. And the publications all thought it was one protein enzyme, but we actually found it was made of 3 proteins, based on what I did. And because we were first, we had the patent rights. In those days, you could patent genes and publication rights, and that really blasted my career big time because it was huge. I won awards after that. I won opportunities to travel to Toronto for 2 years. It was really exciting times, and I realised then that not too bad at these idea things.
Yeah. So can you talk me through the process? So, because it sounds like, they had a bit of an idea of what they thought or where it was meant to be, but instead of that, it sounds like you kind of were like, okay, you know what, I think it might be different to that. I've got a hunch or an intuition, and I'm going to work with that and then come back to it and looking at through new eyes. And I feel like most innovation and most good science actually is that, right? It's people who don't get stuck in I guess you could almost say like the tram track of what's been going on, because there's obviously diminishing returns if you keep going a certain path in science. You're reducing your surface area to the unknown if you're constantly going the same way, and sometimes you need to look to the left or to the right, or potentially backtrack a bit and try a different path, which then opens up an entirely new frontier. So I'd be curious to know, like, what was your process with trying to figure this out? Because it would have been, as you said, it was analog, right? Like for people like myself, the first thing I'll do is go and look online or check out GPT. Like we have like the Library of Alexandria at our fingertips. It's a very different way of cognitively processing information. You're much more of like a synthesizer of information that you probably don't know deeply. So yeah, I'm curious to know how different that was for you back then when you're trying to figure this out. Like, what was your process?
Process, follow what's known first, get an understanding of the situation, and then just allow my mind to go free and start thinking up ideas. And that's when I first realised that I had a mind where ideas just went flying through all the time. I didn't realise till later it was my daydreaming method. But I have ideas just flying through the back of my mind all the time. And ones that I liked, I stopped thinking and pulled it in. So I was very, very creative. It was different way of thinking. And got the approval of, as I said, of the powers to be to do this. Because I was halfway through my PhD at this stage and I hadn't really got any results. So I was under a bit of pressure to do that or change projects. That was also in front of me. But I believed in what I could do. I knew I had an idea. And so we essentially made 3 Velcro materials. And I sort of put what we had through put the pig liver stuff through it, and watched what happened. And one of them stuck like glue. And when we finally got it off, it was in its perfect state. So it was really just allowing my mind to go free and be creative and trust myself, trust my ability, trust my instincts. And that was really the basis of my science career from then on, and still to here as well.
Yeah, okay. So, and just so that I understand, so when you say like a Velcro-like, is that you gave that a certain charge, or you had certain molecules or things attached to it, and then you run like a fluid through it, and then it's picking things up? Is it kind of like that?
It's like that. We knew that. So the way this enzyme worked is it liked having peptides. The way we measured it was we had a peptide that was something it really liked, and it transferred, and we had mixed it with radioactive ATP in those days. And in those days, you could eat in the lab, and about 5 metres away was a radioactive laboratory, experimental space with only a perspex screen to protect you. Meanwhile, you're eating it anyway.
At different times, right?
different times. So there was the way it worked was that you incubated your extracts and some radioactive ATP and this peptide, and then you collected it on some special paper that bound it.
Yep.
And you just measure the radioactivity of what's been happened. Has that? I don't explain it very well. The enzyme takes a phosphate off ATP, cuts it, and transfers it to the protein or peptide of interest onto a amino acid called serine or threonine. And then you could measure that. And that's how it was measured, and that's what was published, how it was measured. The lab I happened to work in at St Vincent's Institute of Medical Research, they made lots of peptides for all different series. They had like a library of over 1,000 of these peptides. So we looked at similar ones to it. We screened a whole lot where it had similar activity. And then we attached them to what's called a chromatography column, solid support, tiny little things, and then lined them all up against each other and just put different versions of that extract through it, washed it all, look what came through, what didn't come through, and one of them, it didn't come through. It was stuck on it, and it needed a very harsh treatment, high salt, high almost ethanol, to get it off because it was bound so tightly. And in those days, you'd run things on a gel, and you'd stain it, and you'd destain it, and eventually you see what was there protein-wise, and there was 3 proteins, all in equimolar relationship, which suggests that you had a trimer of proteins, which was not heard of at all.
Interesting. So not only did you discover ATP kinase, which is like a big deal, like...
Yes, there's a conference on it every 2 years.
Yeah. I mean, I learned about this at uni. You know, not once do you learn who found the thing, which is so fascinating when you're learning science.
And in those days, there was no mass spectrometry. No mass spectrometry.
It sounds like you did a lot of it more analog.
The database only had yeast proteins in it. It was very young. It was just before PCR. It was really early days. And the only thing we had was an amino acid analyzer, and it took 1 hour per amino acid to come off our sequences. So we would sit all night. Getting 10 amino, and then put that in the database. Is anything? No, not yet. Keep going. And finally, we found it related to a yeast protein, thank heavens. Quickly got the story together, quickly published it, got accepted overnight, as quick as I've ever had something accepted, and won the race.
That's so fascinating hearing that story. And I imagine now as well, like, for you to still be in the game and still doing what you're doing, you would have had to have adapted and evolved your systems or processes for how you do science. I'd be so fascinated to know what has shifted and maybe for the worse and for the better with doing science now versus when you first started.
The biggest thing that shifted is just the admin, the paperwork, the risk assessments, the lack of creativity's gone out the window, I think.
Yeah, with the
ability to be free and do what you want to
bureaucratization
do
of everything.
Think what you want to do, even with animal models.
It is interesting as well, like how much now with things like what cortical labs are doing, we actually might have the capacity to remove the need for animal models for like some sort of like drug analysis and stuff like that. So it is interesting that we're progressing to a point where we don't have to use them. But to your point, yeah, it does feel like there is a big admin burden that a lot of scientists are having to deal with. Like when you look at papers now, like back in the day, I don't know how many authors you had or co-authors you had, but you know, maybe 2, 3, whereas now you look at a paper, there's like 20, 30, you know, and the discoveries don't feel as big or as amazing as what they were back in the day. It's really interesting just noticing little shifts like that. But then I guess, you know, I assume that's maybe what pulled you out of academia as well. Were you aware that there was like the startup realm whilst you were studying? So this was
never heard of it.
Never heard of it.
It was
never
part
mentioned
of universities at all. It was early days, and I left science because I had to. My research funding ran out. The success rate. I could get grants when it was 15 to 20% success rate, which is pretty low. But I had a pretty good track record. When it got below that, I couldn't. All the big boys got grants. It was jobs for the boys, and
Yeah, I've heard it's kind of like that.
I had to go to my minus 80 degrees freezer and then put my arm and pull it across and put everything in the bin, which is devastating, all these samples you've done over the last 8-10 years. And they hand you pass, there's no farewell, and off you go, you're out.
Yeah, it's fascinating how competitive and cutthroat it seems to be. Like, I've never worked there, so I really can't have an opinion on it, but it does seem to sound like almost like bringing the worst out in people, and it's not because anyone wants that, it's just the way that the system is designed, and it seems like there are maybe perverse incentives that make it end up like that, but yeah, so I'm assuming that pulled you out, but it wasn't Utilium wasn't the first place you started. You had a couple of other little side projects.
And before that, a really good bit of advice I got as an exit interview.
Yeah.
The director was, your PhD will carry you a long way. And in my mind, I thought, whatever. But they were right, because the public really looks up to anyone with education, particularly a PhD. They don't care what it's in.
It could be memes.
And I realised that, and I started to use that a lot. And I still use it today. I'm very comfortable in it.
Literally the only reason why I would do a PhD is so that you have the weight of the title at the end of your name. Very useful for credibility, especially if you're trying to be the CSO or something like that for a company. I imagine it was quite useful for you with the B12 patches and is it B12 magnesium?
Yeah.
Was that before the hemp gin? Or
yeah. So I left. Just before I left, I'd been at a conference overseas, somewhere in America, I think, and I saw a little shop selling supplements on a patch. I thought, that'd be interesting. So, because I get a lot of brain fog, it's really hard to get rid of it, and I tried a vitamin B12 patch, bought some, and my fog went gone completely. I thought, oh my God, that's a game changer for me. Wish I had this earlier.
Yeah, I have the same thing. Yeah,
and I thought, we should have these in Australia. So when I left, I just started learning about setting up a business, an ABN, learning about e-commerce websites, and sort of taught myself how to use Shopify, and got on there and started selling these patches.
So it must have been relatively recent, like the last 10 years? Okay, cool. Yeah.
About 10 years ago, 10 years ago I started it. Just started selling a couple of vitamins. Then started looking around, getting confident with Alibaba and negotiating with Chinese and getting stuff sent through without getting picked up in ways that they perhaps should have been.
Yeah.
Anyway, so that was my introduction to it, and that's been pretty handy. That's kept me going. No matter what else I've done, I've always had that, because it's always paid my rent at least.
A little bit of supplementary income sort of thing.
It is. And one of my things about me is I'm really passionate about making a difference in health. That was my goal when I started science. And now I'm in startup mode. I've extended that to the planet, make a difference to health and the planet.
That works for me.
So shifted everything across now, but it's always been my mission to do that. So Patched Up, it's called Patched Up. It's not about making money. It's about making a difference in people's health. Someone messages me and says, David, I love your B12 patches, but I need to use them every day and I can't afford it because I'm on a pension. I go, no problem. And I just hardly make any money for myself, make sure they have what they need. That's who I am so Patched Up was first. And then at the time, it was just when cannabis was starting to come on people's radar, and hemp seed oil was just about to be approved as a food. 2017 or 18, I think. And there were a whole lot of meetup groups about it. I started going to them, and I met a guy who had saved the domain name, The Cannabis Company. And we chatted, and he said, how about I give you a few shares and you be my head of innovation? I thought, sure. I knew nothing about it. No AI to help me, but got into PubMed and things and learn about it.
Got the PhD, so.
And it didn't sell cannabis. It was building a brand towards when it was allowed. But in the meantime, hemp seed oil was there, and I found out I was pretty good at innovating then, because I started selling hemp seed oil for pets. That was the biggest seller by country mile. Reducing inflammation, helping everyone love that, and we soon got a marketing going as well, and it really just started going really well.
Mm.
And then there was discussion amongst the three of us about making a gin, maybe just with hemp gin. And at that time, I'd just come across these things called terpenes, which are essential oils that consisted of one major terpene and a whole stack of about 20 or 30.
And every plant has terpenes, right? Like it's the volatile organic compounds that they might release to communicate or
protect themselves,
or heal themselves. Cool. Yep.
Yeah. And so for us people, we always say they have a smell, a taste, and they do something.
Mm.
And so, there's eucalypt and there's...
I'm big on my essential oils. Like that was what I always thought I would be like a little alchemist, making sort of medicines and extracting oils and stuff. So that was kind of one of my draws to science to begin with. You quickly learn that it's very different in...
And did you have a favourite one?
Lemon scented eucalyptus is probably my favourite smell, but I don't know about the functionality of that per se. If I'm thinking functional, I would usually use like a lavender or something for me. For me, that's such a calming thing. Like you might often see me with lavender in my ear as well. That's just something that I find incredibly relaxing. And then rosemary sprigs as well for just like alertness or working. So I would use those. And then essential oils wise, I just knew the importance of when you walk in a forest, so breathing in all the terpenes. So I actually, in my office, I actually have like a little nebulizer that has like cedarwood, sandalwood, fir or pine, and then I'll throw in a couple of different flowers here, there, and everywhere, based off sort of like that principle of like when you're in a forest, your body relaxes, you actually can think more clearly, and it is beneficial to your immune system when you're breathing all this in. So I guess, trying my hardest to emulate that in an office environment. And I find I always feel a lot better.
You're the only person I've ever known who knows that.
Huh.
In Japan, they use as a health retreat. They put people through them. And
Forest
when they come
bathing.
out, they're so much better. And that's why it feels good walking in the bush, because the eucalyptus trees are limited to beautiful elliptical. And it makes you feel fantastic.
All the indigenous folks always speak to that. They're like, eucalyptus is sacred. It's medicine. It heals you. Yeah. That's why we use it for every time there's a fire ceremony and a smoking ceremony. It's usually eucalyptus, and it's such
I learned
a beautiful...
about terpenes when I was at the cannabis company, and I started researching how they were anti-inflammatory. But how come I haven't heard about this as a biochemist? And I started looking at the literature, and it up to 1985, and then it stopped. And I wondered if
Nixon
big
stuff
pharmaceutical came in and said, this is threatening our livelihood. We're making the anti-inflammatories, not the plants. And that's when I came into science just after that. So I haven't got any proof to say that, but I suspect that's played a role in it.
It's definitely the case. And I think what's also really interesting about cannabis is the fact that we have our own internal endocannabinoid system, which I feel like is more known now than before. But it's quite fascinating to think that we actually actively release that in our body as a communication mechanism already. And so these things are binding to receptors that we already have in us, which is just a fascinating quirk of biology, how things are reused in a different living system, even if it's millions of years later. And it's just so fascinating that they can still interrelate.
I've got another theory about that too. Because cannabis was dropped, made illegal for a long time, a lot of diseases popped up in that space that time. And I'm wondering if it's because their receptors weren't being utilised by their common ligand that was meant to bind them. Even hemp seed oil was out. Hemp oil was out. So
it was a staple food, and we used it to make. We made sails out of it. Like we made so much of our clothes were made out of that. There was a thriving hemp industry all throughout Europe. It was only as you said. I think it was, and you see this happen quite a lot with commercial interests. Like you can't patent a plant, so there's no research on rosemary or all of these other herbal things that have been used for thousands of years. But if you can extract it and you can refine it, and then you've got something like, is it you or one of those trees that you can make a antibiotic or something out of, but you need to refine, and then you can patent that. So it's interesting how all of these things that you can't patent that have a known effect don't get any funding or any research, or are actively shut down by folks making money out of a synthetic analog or a small molecule drug that does something that is probably not as good as a natural one. But it's an interesting quirk of the
There
system.
were 3 different ways that cannabis could bind with molecules,
Mm.
and they were all vacant most of the time because we weren't allowed to have the material, have the ligands that come in. And I always wonder, what diseases came along because we had those systems were blocked. I think that's another mine.
It is fascinating.
Yes.
It also reminds me of like if you don't expose kids to nuts, they get allergies to nuts. And I'm wondering if there's some level of exposure to a wide range of terpenes, which actually creates a healthy functioning human and immune system. And that in this world where we get most of our food from, let's say, 4 to 10 crops, how much that probably impacts just everything. Bit of a
So when the cannabis
side quest.
thing
But
started to explode again, terpenes became alive again. And terpenes are everywhere. So there's one called myrcene, which is in cannabis, but it's also in mangoes and a whole lot of other types of fruit as well. But because they're in cannabis, they became alive and a big business. People selling terpenes for all sorts of things all around the world, not just for cannabis-related stuff, but all health. People realised and started innovating different ways of using it. Like us, we decided to add one to a gin. And we went up to this distillery in Healesville called Alchemy.
very fitting
It's just
name.
across the road from Four Pillars. And they do some, make some gins for you on the side. And we told them what we wanted, and I said, I want to put in this terpene. Oh, it's too oily, it's not going to work. I said, yeah, just give it a shot. So he gave it a shot, and we went up for some tastings about a month later or 2 months later. And he met us at the door. He says, I was wrong. This is one of the best gins I've ever made in my life. This is going to win awards. And which it did. It's the San Francisco Spirit Awards. So that was our launch into another product. Here I'd gone from a meek and mild scientist to making cannabis gins.
I saw the articles, man. It was so funny reading through some of those, just like the Startup Daily one or like the Good Food. It was really interesting hearing how they'd like framed it all, and it was all, well, because we've got this scientist involved that's very, very legitimate, which was a really interesting.
Yeah, the PhD coming again.
Yeah, seriously, but fascinating. Okay, so that's where you got your taste for innovation, right? Because, it's one thing, like, don't get me wrong, like, you've published, like, let's say 100 papers, right? So very proficient in academia. But it is a very different mindset when you're innovating in a commercial context or in a context where you don't have to just like, do a triplicate set and then retest and ensure that, there is a 99% chance that it falls within a 10% deviation of the, that's kind of all out the window when you're working in a commercial context. So did you find that it was easy for you to make that switch? Like, what did you have to unlearn from academia?
I loved that switch because I was that person in science.
Interesting.
And I was controlled by the rules, particularly the more I went on. In the early days when you can eat in a lab, you could do all sorts of things. But once that changed and got more controlled, then I got controlled. And I was really starting to get more and more limited and more frustrated. So I wasn't as disappointed, I suppose, in hindsight that I lost my funding and had to go and live my passion. I'm a free thinker. I need to have the room to do the ideas, to think about it. I need to be trusted with who I am as Dr. David. That's when I started using the PhD a lot. And that exit interview proved 100% correct.
Mm.
So I'm a natural innovator. I realised that in my PhD. Postdoc I did in Toronto, I was also stuck. I realised I'm really good at ideas. I'm really good at creativity. I'm really good at adapting to things I know nothing about and learning about it real quick and then seeing the applications to it. And being in the cannabis company, that really helped me realise that in a big way. Thought, I'm actually really good at this. And the things I'm making are really successful as well. Like the second gin we made, it had beta-caryophyllene in it, which is from pepper. Has a lovely smell, anti-inflammatories as well. And at that time, the Australian Financial Review came and did an interview with us just before we launched that. And apparently, and then we had a little tasting place too in the warehouse we're at. All these old ladies started appearing, old men, and apparently they've all gone to their rheumatologist who's read the article and said, I found a solution for your anti-inflammatory, it's this gin, go and buy some. So we had hundreds of elderly people come in, desperate.
Desperate for a G&T.
They bought heaps of gin from us because we said it was anti-inflammatory. The Australian Financial Review convinced the GPs, specialists.
That's so fascinating.
And anyway.
But it's true. Like, so a lot of tinctures back in the day, we would use brandy or something like an alcohol to suspend the terpenes, which we didn't have the name for it back in the day. But that's exactly what we did. So, even though my head's going, oh, but it's an alcohol, it's like, yeah, but that's a really efficient delivery mechanism. So that's...
maybe that's why they came. They had that growing up as a kid.
Fascinating. So, okay, and then...
and also the terpenes
were
important because they, where I am now, they are in my mind for the first time and I started learning about them, particularly what they did on the bottom of boats. And that was an idea that stuck with me through other things I did at that time.
Terpenes are related to the bottom of the boats.
Yeah, they're antibacterial as well. And I also learned from a friend of mine who cleaned the bottom of ships as a business, travelled the world, took them out of dry dock, cleaned them all. He told me about how a layer of bacteria land on the bottom of boats first, and then they form this slimy thing around them. And then once they do that, they're like Velcro and attract everything that comes to them. But it's the bacteria first. And while he was talking, I thought, I know some terpenes that might fix that up. So I kept that to myself for the next 5 years before I actually acted on it.
Percolating in the background there.
Yeah, just leaving it there, watching the literature, see what was coming.
So tell me a little bit more about the idea that you had with the bottom of the ships. So you've been mulling over it for 5 years. When did you have that sort of moment of insight of like, okay, I think I have a solution?
It was more than, do I have a solution? Was it the right time for me to do it? Was I motivated enough to start something new? So I had another business in between cannabis company and this one, plant science. And I still had it in my head all the way through that space. Plant science is with a farmer, another academic and myself. And this showed you how good at innovation I am. I know nothing about horses. I made a probiotic for horses to stop them, particularly ex-race horses who are stressed, to be calm in 1 week. And I know nothing about horses. I know nothing about hemp fibre, but a bit of research, it takes me 5 minutes to understand what it can do. And I'd made this great product, which is still for sale for now from Plant Science.
There you go.
But I had the idea all the way through that, and it wasn't till I had a scare with health, and I was in hospital for a couple of months with an undiagnosed pneumonia. As I was recovering in there, I thought, I've got to get out and do this terpene experiment. As soon as I get out, and I'd only recently moved from the hills of Eltham, Warrandyte, to Bayside, then St Kilda, and I thought, I'm close to the beach, now's my time, I'm just going to give it a shot. And I decided to do that. And when I came out of hospital, I'm still pretty sick and not diagnosed properly yet, but I staggered through all the pain and bought some rocks from Bunnings and epoxy resin hardener from a marine shop in Elizabeth Street. And I bought some essential oils on Amazon because I couldn't buy the terpenes because I needed to have a chemical license and be part of somewhere like here.
Mm.
So I picked the one with the most likely chance, and that was oregano oil.
Makes
You know,
sense to me.
oregano was
Yeah,
very
yeah.
strong.
So I used to use that as a herbal medicine treatment for throat infections. So it was really good for.
You don't want too much of it though.
No.
Burns.
You had to dilute it like you'd put like 1 drop in 250 ml or something like that. Mix it around, but it was it's so potent and it actually burns. Like if you get a drop of oregano oil on your skin, it's insane the amount of like. I don't know what is in that, but
It's called carvacrol, and I've got some in the lab. You want to come and smell it?
It's
so strong. It's 90% of the essential oil. It's carvacrol, and I've had my eye on carvacrol for a long time. Been published in biofilm journals, but never really been used properly. Thought I'll just give it a shot. So I got these rocks. They weren't that big, and painted them in the backyard where I was at. And in one of them, I had a guess percentage, 3% essential oil in it, and the other one with nothing in it. Naively went to along St Kilda Beach, off Middle Park, is a thing called a groin. It looks like a pier, but it's concrete, and it's actually to help. It's got storm water coming out. It's just protecting it. And on one side of it, it had a whole bed of mussels and stuff. I put my 2 rocks, tiny rocks, on this bed of mussels, unaware of tides and things, just thinking it'd be safe. For 93 days, it was safe. And I put it in wintertime, or just as winter was coming. Again, I had no idea about. In hindsight, I thought winter was a windy time, but it's really still in the bay, fortunately. So every time I come back and check my 2 rocks, they were still there, amazingly. Tides kept coming and going. And I started watching. I learned about biofilms by then, which are communities of bacteria covered in the slime. And I noticed the slime come along after about 20 days, or maybe less than that. Maybe 2 weeks. But that's what they're talking about. And then I started noticing brown spots, and then I started noticing green spots. And then I put it in the water, and up grew this 3-dimensional biofilm on this rock. So I learnt about biofilm hands-on watching it grow. And then one day I came back at spring. The spring tides had come in and gone. Somewhere in the ocean. Still doing well, I bet. And the control... And that was just with the oregano oil in it. It had nothing, absolutely nothing. 93 days. I thought, wow, imagine if I had the pure stuff. So that really got me into biofilms and understanding how it did. And I thought, I need to start a business in this. So I was meeting with my step sister, and our parents were elderly, and we need to get them into an aged home. So we're chatting about that, and then she said, "So what do you do?" I said, I'm out of work, but I've got this I'm really good at ideas. For example, and I talked about this example. She goes, you should meet my friend Rob Gell, who, for those that don't know, was a legend in the 90s when he did the weather. He was one of the hottest celebrities in Victoria at that time, and people would even use his name as a verb when someone says, it's going to rain tomorrow, and the other person says, who are you, Rob Gell?
Yeah.
And he's now the president of the Royal Society of Victoria, Water Australia. We had a chat, and he says, I'd love to start a business with you. Let's come up with a name. So sort of casually how it started, Utilium started. And Utilium, it's a bit of a mistake. He meant to find a Latin or Greek name that meant coatings. And he chose Utilium. He thought that was it, but it actually is better because it stands for utility. It does everything.
Mm.
And the way it's worked out, it's actually a better name than a coating only. So anyway, that's how Utilium started, and I potted around and I got a bit more confident, and I started painting concrete and other things, and really learning about biofilms and biofouling, buying lots of hands-on learning.
So for those that aren't super aware, could you kind of go into a little bit behind biofilms, biofouling, what that is? Because I think people might be like, I have no idea, but then everyone will probably know. So have you got any like pretty concrete examples of where you might see that day to day?
Firstly, it's a bacteria love sticking together in water or on water. So under the water or on a wet surface. You might have a wet surface around your house.
Why is that?
That's a good question.
Is it the movement and access to?
No, I think it's the way what they do once they're together is they send some signals out. We're big enough, and they
It's like the core
create this
sensing.
slimy cover all around them. And I think it needs the water for that. And once they've got that around them, they are indestructible around the world. And cost the world $2 to $3 trillion a year in damages.
With a
health
T.
Industry. Yeah.
Trillion. Wow.
Trillion. It's a really huge issue. And you may see it in your washing machine, in that little rubber thing when you open the door. If it's anything black in there, that's a biofilm. Need to get rid of that. That all goes through all your clothes. Might be funny stains on the bottom of your shower or your pink you can't get rid of.
The pink. Yeah.
Pink
So
or orange.
yeah.
I've got an orange one in mine. And
That's
nothing
from the
will get rid of
soil,
them.
isn't it? It's a soil bacteria that, to your point, once it's established, good luck.
And it's living in the drain. They're biofilms. They're on chronic wounds. You might know someone who's got a chronic wound that can't be healed. It increases antibiotic resistance by 1,000-fold once that's on someone's skin. It's a really huge problem. Clogged pipes, bottom of boats, water purification, food industry, agriculture.
Hospitals then
hospitals
as well.
everywhere. Hospital sinks is a disaster. Anywhere in a hospital can be a hot zone.
Yeah, no, it sounds like I know that they're everywhere, right? It's so fascinating. So if we were going to think about it, like a biofilm's more like an infrastructure or
It's
an
like an organelle.
ecosystem.
I treat it like a mitochondria
Interesting.
or something like that, because it's surrounded by like a membrane. And there's a lot of signaling, a lot of stuff going backwards and forwards. And inside, it's an active unit. And this would be heretical to microbiologists, but the bacteria are irrelevant once they're in that side. I liken it to glycogen, the molecule glycogen. I used to work on glycogen in exercise research. And it's got 50,000 glucose units and a few proteins. So we never once talked about the glucose when it got reduced with certain exercise or disease or changed, or about the proteins. It was all about just the glycogen. For me, the biofilm is an active unit, and everyone does their part, but it's not about one part like the bacteria. And I think that's one of the issues with the research currently, why nothing happens, is it's treated like the bacteria are the most important. But
Rather than, so this is a really fascinating point around like reductionist materialistic approaches to science can be limited in the way in which they perceive reality. So rather than seeing, which is interesting as well, because you'd think that we're so used to in science seeing that it's a part of a system, like we call it the endocrine system, this system. But it is funny that we still sometimes fail to see how things can come together and there is an emergent phenomenon where the whole is worth more than the sum of its parts. And specifically in the biofilm space, it sounds like that's a real thing that you have to kind of comprehend and almost see it as, is it something that is beyond and above? And from what I can tell as well, they almost communicate almost like it's a brain, a hive mind, right? Like they can send signals where something will fire off here and the entire, I guess, colony or collection, the swarm.
Yeah, they have a thing called dispersion. So their life cycle is find a surface, accumulate everyone together, and fungi are in it too sometimes, and put the slime around them. And then if they sense a surface nearby,
So a SCOBY is kind of like a biofilm as well.
yeah, if they sense a surface they want to colonize, or if they're too big for not enough food for everyone there, or something like that, they will send a signal to the slime and it'll open up a door and a cohort will go out, bacteria will go out and do whatever they need to do to find somewhere else or colonize another space. There might be a boat nearby or a rope or something they've seen that they want to get onto.
So it is like a very much a collective intelligence.
Yes, correct. And then it closes up. The thing about the dispersion is they're vulnerable for 2 to 4 hours because they're open this time. No scientists know how to open it up. Well, not many anyway.
So now that is interesting because this is where you've tried to look at it differently, right? So instead of targeting the bacteria, you're gone, well, what happens if we can nudge it rather than kill it? So this nudging concept is something that I'm really curious about if you want to speak more to that.
We're looking at behavioural change
Yeah.
of the biofilm, which is completely new. I just went to a biofilm conference 3 weeks ago and no one mentioned that at all. And being a biochemist, so biochemists, we look at proteins and enzymes and systems and organelles and stuff. I'm coming into this space, I should say, with no microbiology training at all. I've never published in microbiology or bacteria. I hardly ever used it in my career. So
Something
for me,
to beginner's mind though.
I'm looking at this as an organelle. I'm looking at this as a whole complex. I'm looking at it differently to other people, microbiologists who grew up in that space and how they work. They see it as bacteria only, not seeing the whole thing as a complex. And I like seeing things as a whole complex, how a cell works, how mitochondria works, how a nucleus works.
That holistic approach, I think, allows you to gain insights which can seem almost magical to others who might be stuck in a bit more of a reductionist approach. But to me, it makes sense. That's just the way that I try to look at the world, is through like that systems lens and acknowledging the interconnectedness of everything from right from the smallest living unit of the cell all the way up. And then you can even say that even smaller than the cell, there's still entanglement and things like that going on at a subatomic level. So it's all interconnected and interrelated, and seeing them as separate or only focusing on parts without it being attuned to the whole seems like most of the best science is going to come from actually trying to put these things back together and look at them as you're saying, as a complex or as a whole, which I think is super cool. So okay, great. So you're looking at nudging their behavior. You're looking at shifting them so that they.
I'm looking for weaknesses in them that are not known scientifically. So although there's probably 100,000 scientists working on this, for me, they're relevant to me because what I'm doing is different. I know what's doing different to them, so I don't feel under any. There's no pressure to publish or find stuff, but I'm looking at ways to nudge them. Do they move? Is it a way they open up? Can I stimulate that dispersion? Can I tempt them out of their fortress because I've knocked on the door the right way and I've got the key to open their
So it's almost like
the outside.
Semiotics, like you're looking at communication. Like, can you communicate with this unit and convince it to behave in a way that
trust that what I'm doing is safe. And we've found a way to do that, I think.
Nice.
Using,
Yeah, I'd love to go there. So I think that's a perfect segue. So you mentioned before that you put these rocks on a bed of mussels. And I know that the story goes that after you'd been experimenting with the oregano, you had a bit of an insight or a realization about, let's just say like a lot of marine animals that you will see, even when they wash up on the shore, they're like pristine, like the shells, not all shells, some shells, which, they literally are designed to attach to everything. That's a different story. We're not talking about them. We're thinking more like crabs and crustaceans, and they always look immaculate, even if you pull them out of the bottom of the ocean, which is fascinating because they're filter feeders. So they're getting exposed to all manner of gunk, but they always look perfect. So that was kind of a bit of a moment for you, like a bit of an insight.
that was a big insight. That was a big insight just before I moved in here, actually. I completely changed the project I was going to do. The week before I came in here.
That's
I called
interesting.
Andrew and said, I've got to change. Is that okay?
Well, thanks for letting us know.
I wasn't expecting it. I was going to do the terpenes on the bottom of boats. But I do like challenges, and I do like looking at big problems and finding solutions to them. I like the challenge of that, and the importance it is to health. And I'm driven by health, and now the planet as well. So I'm looking at this. So I've since moved even closer to the beach to Port Melbourne, and I'm about 10 minutes walk to the beach. So I walk along the beach, and I'm looking at shells going just after Christmas time a couple of years ago. Well, how come these have got no biofilm on them? Just suddenly realised it. What about scallop shells and prawns and stuff I see at the South Melbourne market? That always looks pristine. How do they do that? And the dogma is it's chitin, molecule called chitin, which if you put in the lab and treat it with acid, it's going to work. But that's not going to happen in an animal. It's just a structural. It helps. It's their spine basically. It gives them structure and strength and shape. That's all it does. It does not do anything to protect them from slime growing on them.
Well, it couldn't, because there's obviously so many other chitin-based marine animals that can get slime and gunk on them. So it might be a necessary component but insufficient, right?
So but that wasn't it. So so I have to give this a shot. Worth a shot. Got this great place, CoLabs, to try things. I've got AI that's appeared into my life with instant access to all knowledge, as long as I ask the right question. And so AI and I started doing some experiments, and the thing I've only just realised is for the first 9 months, I got all these results. If I was in a laboratory, a research laboratory with a big group, I would have been told to change projects within the first week, because I was interpreting the results the wrong way. But because I'm such a positive thinker, and I had an idea in my mind that something from shells was going to change the behaviour of these biofilms, I believed the opposite. I just believed that whatever I saw was due to biofilms. And for example, I knew that biofilms had a symbiotic relationship with vegetation, like the green stuff you see on rocks down on the beach. It's called sea lettuce. They have a symbiotic relationship. Bacteria make vitamins for the...
Makes sense. No different to terrestrial plants.
And the sea lettuce give the biofilm somewhere to live. And so I started grabbing some of that from down the beach and putting in this tiny container with some seawater in it. And if I saw some bacteria move more than 5 centimetres, I blamed it on the shells I had underneath. But it really, in hindsight, I now know that biofilms on sea lettuce can sense things 20 centimetres away. And I had it this far away, 2 centimetres away.
So they're trying to move away from the shells.
Well, they're trying to take over another surface. And they go, oh, gee, that one's a good one. That lid of that container, I'm going to jump on that. But I assumed it was coming from the scallop shells that are underneath the sea lettuce. So it's scallop shells, sea lettuce, a little bit of water, and then a lid. And because I saw bacteria, I go, yes, that's worked. But I now know that, no, it didn't work. It was because of the physiology of so biofilms, don't forget, they love to sense a surface. I had one 2 or 3 centimetres away from them, which they jumped on like they would if there was a rope near the bottom of a boat. But I assumed it was
trying to escape from the
shells. And I conducted my research that way all year until I started asking some of the newer AIs coming out hey, I got all these results. What do you think? Oh, it's contamination. You've got bacteria in there. You're not seeing anything. And I realised that I'd been interpreting all my results the wrong way. But because I was so driven to find a change in behaviour, because I was so positive, I was still on that pathway. And all I needed to do was tweak a couple of things, and I was on the right track. Hope that all makes sense.
No, that does. It's almost like the irony of like so much of science never uses a scientific method. It's intuition, and then you backwards map it on and go, yeah, of course I did this. But it's kind of ironic that at this point you actually had to go back to using the scientific method rather than, you had to check those assumptions at the door, and you were lucky that you had an AI that you could utilize that could help you check that.
Gave me a different opinion.
Yeah.
ChatGPT was very positive and supportive of me.
Yeah, but that's a little bit
agreed
sycophantic.
with everything I said. And when I went to Gemini, it was a complete opposite. It was sarcastic.
That's just Gemini, so right.
Which I needed. I needed that because I'm working on my own. I've got no one else around to talk to. I need literature and stuff like that. So, and I was working in your area. So just let me explain to people, what I had created was a really cool model in exploring biofilm behaviour. And it seems to be a first. No one else has done. I can't see it published anywhere. And that's the idea of putting a little bit of sea lettuce, maybe a gram, that's been taken from the beach, lowish tide, and putting seawater in as well, because such a good buffering capacity. Has to be a really long tube, so I found these great 100 mil long test tubes online. And then I treat shells in different ways, different temperatures, different conditions, combination of different things, and I let them soak in water for a couple of days, and I take that water and I put them into these test tubes, and I look for any change in behaviour of the biofilm. And once I got myself right and corrected all myself, got rid of all the bacteria, I started coming in the morning and seeing a cohort of bacteria sticking to the surface of the water in the air, hanging down. And I started refining that more and more, and I was confident I had the right condition. And it was causing this thing called dispersion. So even though I failed in my interpretation for about 8 months, I did create a good model along the way. That was a good idea, and that was an idea from the start of my experiments. And I just had to adapt the way I was doing things. Still positive, no, all right, done that wrong, but I'm still know I'm going to do it. And I did find something along the way, and I've since taken that water from the shells after a certain condition, and I've done some biochemical analysis on it. And they still need some mass spectrometry on it, but it's looking very, very new and positive in how it opens the door. I found something that mimics the key to open the biofilm up and let some bacteria out.
Fascinating. So it's almost like that communication piece we were speaking of earlier. So I'm curious, what's it been like, because obviously, we welcome you in through the Impact program. We believe in what you're doing. We're trying to support you making this happen. What's it like working in a space with very limited funds? And I guess, as you said, you didn't necessarily know much about the space. How has that shaped your way of being and your way of doing? Have you noticed it's been a big shift, or I'm just very curious.
Yeah, it's been a huge shift, having no money in particular. However, it's been a good shift, and I think it's really important. This should be important for all impact members, is to use the tools that you've got here. Don't go out to too many other analyses, particularly anyone doing sort of biological like I am, because the only equipment in the lab, apart from just general things, was a spectrophotometer that Diagnose put in there so they would get, cheaper rent.
Yeah.
Or something like that. For
Thank
everyone
you,
to use. Andrew managed to get some special plates for me from Merck for free as a favour. those two things, if I was on a big lab, I would have taken that stuff, I would have put it through a mass spec, I would have missed everything. The gold is hiding in the detail in what I see in the spectrophotometer. And if I hadn't been limited as I am, I would not have found what I found. It's really unique, and I found 2 needles in a haystack that stick together. I would have missed that for sure. Looking at a mass spectrometer or sending off to Monash to get looked at, nothing would have come back that would have stuck out to me. Because I've had to really analyse using the analytical tools, which you would have done back in the day before mass spectrometry. This is old school fashioned chemistry and biochemistry. Looking at the only data you've got is by shining light through something and seeing how it behaves. And is there a fingerprint of what it does? And I found that. And so I'm very grateful I had no money. I'm very grateful I was so limited to basic tools, and that's why it's been missed, I'm pretty sure. No one's found it because they just crush up some shells, treat it with a few things, put it through a mass spectrometer.
That's an
There's
interesting
a million
framing.
things. They can't see anything different. They got no idea what they're looking for. And I noticed something unusual when I saw this spectrum. I saw a little bump appear where I didn't expect proteins to behave, and then I found out it was a nucleotide. And I thought, what's that doing there? They're the backbone of DNA, and they're like what I worked in AMP kinase. They were nucleotides. What's going on? So I've since tracked that further down, and I found out that the nucleotide, whatever it is, also wherever it goes, a peptide goes with it. Whatever I do to it, I see 2 messages, 2 fingerprints. One's a peptide, one is a nucleotide. So they're either joined together,
Or one
or they have a really high.
chaperones
They're like
affinity,
like either side of a magnet. They stick to each other like glue through some other ion exchange or something else like that, or they're joined together, but they go together everywhere. I would have missed that in a mass spec. I'm so great now. I can go to the mass spectrometer and say, "Here's what I'm looking for," and hopefully it'll be a lot cleaner anyway because I've isolated it better.
Yeah, it is interesting how some of the best innovation happens when you don't actually have all the tools and things, because it forces you to think outside the box, or you have to really start questioning from first principles. And I think, it is really important, not just for impact members, but for anyone doing innovation, is it's like, what's your easiest safe to fail experiment where you can try and validate this without spending so much money? Because so much of science, at universities, you might get a $10 million budget. So you go out and buy a $5 million piece of equipment. And then every time you have to run that, you need 2 PhDs running the equipment. And then the reagents every time are $5,000 to run the reagents. But in reality, you could probably use something that is 80% as accurate for $2. And it's like that shift is such an interesting thing to think about, like, especially if we're going into a future where there might not be as much energy or resources, depending on how like fusion and let's say a whole bunch of other things go. But, if the Strait of Hormuz closing down and us losing access to a lot of fuel is anything to go by, we might have to look at exploring a world where we're using less energy and less resources. And it doesn't mean that you can't get the insights, right? It's just that you have to really try and show up differently and think about things in a different way. And there you go. Who would have thought that not having the equipment that you needed was an advantage working in CoLabs versus an academic environment?
Yep, and why everyone else has missed it.
Are there any other, while we're on that topic, is there anything else that you've, how's your experience been? Have you enjoyed it? What are your thoughts with being a part of our community? I'm curious, yeah.
I love being part of the community. I love the lab space. One of the pluses of doing something like this is in a scientific department at a university, lots of people are doing very similar things to what you're doing. Can't talk about anything. It's too dangerous. People pinch stuff because it's such a dog eat dog.
I find that so fascinating.
10% success rate grants. You're driven to have the little edge on everyone. Anyway, here I'm really enjoying being the only one doing that. What I'm doing, I'm really free to talk about with everyone what I'm doing, what they're doing. I'm enjoying that community. I'm learning, watching Diagnose grow and some of the other businesses grow and what they're doing. So I like what you guys do about the way you really encourage us to grow, different things you talk about, your themes of particular materials. It's a really, I like the architecture, the way you've created the building. It's so welcoming and calming when you walk in the door. You know, it's not white everywhere. It's really, I really love it.
Nice. Oh, it's good to hear. And I know that, the not white everything everywhere is a big thing, and I apologize about shining bright white lights in your face.
That's why I'm wearing the glasses.
Yeah, I know. They help with the migraines, but you also look like a cool biohacker. So I feel like it fits the bill.
Yeah.
You know, it works for me. Nice. Yeah. Well, thanks for that. It's been like so many people just in the community. It's really nice hearing how everyone really enjoys everyone else's company. And it's such a diverse mix of people. You know, you've got 22-year-olds doing stuff who haven't gone to uni. You've got, you know, postdocs, 10 years. Then, you know, so it's so nice having such a wide range of people and hearing how everyone can complement and support one another and then come to ideas or things that you might not have been able to find by yourself. You know, we often speak to the thread of transdisciplinarity and I guess polymathy as being things that we feel are really important for innovation, being surrounded by different thoughts, ideas, ways of being, seeing, doing. And it's just nice to hear like that so many people resonate with that and find that as like a really nice part about being in community.
Yeah. Yeah, no, it's really, I'm really enjoying getting to know the Diagnose team and quantum brilliance and, you know, people I would never associated with in the past in a university setup.
Good. I like that. This is going to be a fun one. What's something like a belief that you held 10 years ago where now you're like, nah, it's pretty much the opposite?
I think for me it's my belief in myself as a scientist. I am really good at what I do, but I didn't believe that when I was in science. I wasn't allowed to really think that either.
Why? Why is that?
It's in a narrow. Had to work whatever you wrote a grant on, you only could work on that, and whatever within your skill set as well. And I discovered I was really good at what I did. If I had those skills now and went back into the lab, or I would have had a different trajectory, different career. I'd probably still be there, which is not a good thing. I'd better to be out of there, I think. So it's the belief in myself. I'm really good at what I do. I'm really good at innovation. I'm really good at thinking left field easily. Just need to know something for 5 minutes and I'm out left field and I've got ideas for people and myself. So I think that's been good and that's helped my personality as well and my relationships with people, just having that inner confidence that I'm really good at what I do.
Definitely makes a big difference, and it's interesting the bell curve of like people very early on, if they haven't been completely broken by academia, they have that. And then there's this big part where people are in the middle of like the whole stay in your lane, don't do anything else. And then there's, you know, the other side of the bell curve, which is like, I know all that stuff and I'm going to be creative and do what I want. And it's really interesting. We find people either at that very start or at the very end, you know, that's where a lot of the innovators that we're having come to us come from. But I think that having gone through that journey and having the wealth of knowledge of being in academia and then having the ability to phase shift into innovation, you know, you can bring that wealth of knowledge with you, but not everyone has the capacity or the capacity for self-reflection to be able to critique or question what patterns and processes from academia no longer fit in this context. But it feels like, like maybe because you actually lean on your intuition and your creativity a little bit more, that's kind of allows you to break the mold of what a conventional materialist scientist would be.
I've also learned how to access my subconscious through daydreaming. That's been something that's really developed in the last 3 years. I always knew, and a lot of people too, a lot of good ideas come through daydreaming.
Most of the best ones, man, like Da Vinci, Einstein, Tesla, all of them used to use daydreaming or the liminal zone between asleep and awake.
Yeah, and holding a rock and letting it fall to the ground. And then
Yeah.
wake up. Yeah.
Yep. Yeah. And then that's that flash of intuition and insight. So I think you're in good company.
Yeah, and I'm too, and I was, good to read those stories and realise you're not going mad doing these things. But
There's nothing wrong with being a little bit mad.
I've now learnt to just create a story in my mind, usually about what I'm doing and some sort of fame attached to it.
I mean,
how great I am, and
that's only human.
And then people saying things about me.
What was it? The Nature paper? You can see the Nature publication.
Yeah, I think about the Nature paper, amongst other things.
I'm waiting for it.
Might be too. I think about those things, and then people around that might say something. You're amazing how you found that nucleotide did this, and then I stop. Oh, that's a good idea. I write it down. So that's how I get my ideas. Through daydream, like even this morning coming here, I was thinking about dip biofouling on glass plates, and if it could happen, and could you do it? And then I was thinking about daydream about fish tanks and interact with different people. And someone said to me in my daydream, "I'm so glad you got that stuff from prawns, because when you put it into water, nothing happens, no slime." And I suddenly stopped. I was driving, right? Gee, that's a bloody good idea. I'm going to try that. I'm going to get 2 stiff fish tanks at home. One's going to be control, and one I'm going to put my prawn stuff, and the fish will survive because they're used to it with the prawns. And that's how I work. Then I put things into action as well. But I get a lot of ideas just by relaxing or going for a walk, listening to music, and allow my mind just to go free. And, in the past, you might have been taught, don't be lazy, you're just dreaming all the time. But it's not. It's a really strong strength if you've got it, the ability to do that and trust what you hear. And the minute you hear what you're after, sometimes I'm not struggling thinking of something or got a blockage, I'll go for a walk with some music. I'll have a daydream. It'll come to me, that's it. And I'll turn around and come home.
And sometimes it's a really quiet voice. Yeah. Sometimes you have to really be tuned in to listen to the whisper. But I resonate with what you're speaking to. I find that so much of my best ideas or things come from when I'm not focusing on it. There's a lot of learning science behind this, like the focused mode of thinking versus the diffuse mode. And that when you are in diffuse mode, it allows your brain to facilitate connections between different areas as you're cataloging, things that you're thinking of going into long-term memory. What do I associate it with? And that's shown that's the most effective way for an insight to happen is when you're going for a walk, because it's like a moderate level of exercise, which happens to, just get the default mode network a little bit more preoccupied because you got to walk. And then that's when these sort of things happen, which is super fascinating. I think it's great to hear you speaking to this because it somewhat feels validating as someone who was very interested in science, but also felt that just focusing in that specific one way in which we're taught didn't really feel like the whole picture.
I daydream into the future a lot too. Want to see where it's going.
What helps with direction, you need in a way you need to have a vision of what you want to be orienting towards. Not to say that you're ever going to get there; it's a final destination, but I find that is what can allow you to be pulled through in moments of duress or stress or why the hell am I even doing this?
Pulled through is a good concept. I'm a big believer in energy, and if you're positive and grateful and believe in all those things, that energy can pull you forward. And I feel that a lot. So I'm at Utilium stage now, I'm thinking of patents, I'm thinking of finally analysing what I've got, thinking of funding. And all my daydreams are around those spaces right now. And I don't control that, just my mind's on that space. And so I'm getting ideas out of that and what feels good, what doesn't feel good, what ideas, which way should I go, what should I invest my last bit of money into.
So speaking about future gazing or future casting, then what, like if Utilium works at scale, like what changes? What happens?
Changes at scale will make a lot of money, I think. Be worth quite a lot. It'll make a difference in at least one industry. When I went to the Biofilm conference, I was unsure whether I should go in where I'd come from, go into health with what I've got, knowing that it could take 20 years to get to market because of all the clinical trials and stuff.
Or marine
or go into industry, which is just straight away. And I came away with a pretty clear direction, go to industry, just to leave all that stuff behind. If someone wants to buy the company and do that sort of stuff, because they work really well in chronic wounds, it's no doubt it'll knock on the door and open it up, expose the bacteria to antibiotic treatment. But someone else is going to have to do that. Or medical devices going into hearts and people, you could stop it also growing on that. But industry is just as needy for it, like blocked pipes everywhere. I've even cleaned out one of the
Thank you.
pipes here in CoLabs that was smelling of sulfur
Yeah.
bacteria. I've tried my stuff on that and it just clears it and it's kept it clear. There's that, there's water purification.
It just makes sense as well. Like, so, you know, health might be something that you're truly deeply passionate about. You know, like you mentioned before, like you fell into this through health. And I think the health of the individual, health of the environment, health of the planet, like they're all, to me, intertwined. But it makes sense that that's where you're thinking. And I think the way that I look at it is like, if you do well in industry, you might then be able to have the funds to be able to then look at exploring a project in health. So like we always say, it's like find the way in which you can look at generating some form of income to begin with, ideally without having to raise any funds. Can you do a trial or a practice run with industry where there's a real challenge that needs to be met and that your tech or thing can make that happen? And then using that to be able to fund future research in other areas. Because with a platform tech like this, you know, there's marine, there's healthcare, there is other areas of industry like weather stations or food.
Food processing is $350 billion a year around the world, problems with biofilms on processes and food recall and processing. It's huge in there. There's no solution. So you're right, and we're definitely going to head that path. In restaurants even, they have blocked drains because of so much nutrition going down the sink. There's bacteria everywhere clogging up things, and they use caustic, terrible things to go down because they have to clear it to keep functioning.
So terrible for the environment, right?
We could stop that with a natural thing. So I'm thinking that way, straight away. We've already got the feelers out for some restaurants with people we know to see if we can trial it. And if that's the way it starts, Sam, then that's fantastic, because that's making a difference to the planet, which is part of my vision, is stopping that, and word will get out, and then we'll try other things. And I know it definitely works well in water things.
Yeah. What I find fascinating about your approach to this as well is like, it's like really grounded in, I don't know if you're familiar with the concept of biomimicry, but that's fundamentally what I'm hearing you say is like, so it's quite interesting hearing you speak to that. It's like, how do we mimic this process in nature? How might we work with, shell waste or other things to be able to bring this about? But what I think is really interesting is it's less about like a domination and more about collaboration in a way with these microbes, and because you can see like us attempting to dominate, command and control nature at the microbial level has led to antimicrobial resistance, all these other crazy things that have essentially meant that antibiotics, the ones like the first gen ones, probably going to be somewhat functionally irrelevant in the next 20 years. So we can't just keep taking that approach, you know. And the way that I'm hearing you speak about this is it does feel like a very different way of looking at how we might work with rather than against these bacteria. I wonder if there's anything there to that, I guess, way of seeing that you think is, unique to your approach.
I think the biomimicry for sure. Being able to manipulate these, direct these biofilms to go in different ways. There's even even for good, there's a slime that's on the outside. There's businesses actually making biofilms to access that because it's a really good material. It's really different. It's really strong. It works well with different platforms. We could do that easily as well. I'm actually making lots of much of that material at the moment with the model I've got. So I could see that happening as well. With antibiotic resistance, just to diverge from it, the toxic stuff going down sinks. That's not only bad for the environment, but it makes the bacteria antibiotic resistant to it, toxic. Yeah. You could throw it in their face and they'll laugh back at you, and you'll be able to stop them and say, you're making a worse type of bacteria and wrecking the environment.
It's like accelerated directed evolution.
Correct.
It's this classic short-termism and like thinking mechanistically about everything. Again, I always end up going back to this because it's stuff that A, really shits me, and B, I don't really get why people constantly think like that. But it is a fascinating thing to think we just can't do that anymore. We have to try and find ways to work with rather than against.
Just on antibiotic resistance, I've got my eye on that too down the track. So to find a good microbiologist. It doesn't work in university.
Cool. Well, we can definitely put the feelers out for that.
What the biomimicry will also do is to sneak into the guards of antibiotic-resistant bacteria and maybe change the messages that they're receiving and drop their guard, which will make them open to current antibiotics, which their guard is up now and they can't get through. So, wouldn't mind another nature paper or two doing that.
Fair enough.
Yeah, anyway.
What do you reckon is the most how do I frame this? Well, yeah, what do you reckon is the most underestimated or underexplored application for like biofilm science right now? Like out of those ones that you've spoken to, where do you reckon the biggest leverage
one I
point?
Mentioned is the dairy farm covered mastitis is a biofilm. Affects the udders. They're completely ignored.
I guess they're not a human and we don't see it.
Agricultural setups have biofilms. It's in the tank, all their water tanks. It's in all the pipes that go and feed all the plants and everything, growth. And they also have antibiotics in them to sometimes keep the bacteria away. So there you're building up the antibiotic resistance and you're contaminating
pretty much everywhere.
Making it worse for people when they eat the food as well. So those sort of areas just get completely ignored.
Sounds like you've got a big task ahead of yourself
You've got to be selective on where we go.
Well, that's the issue is you've got like a near infinite, like this is the thing, like I don't know if you've read David Deutsch, Beginning of Infinity, but it's like when you do create like a scientific discovery like this or explore a certain area, it's the beginning of people being able to do more research, and people could do a whole lifetime's work on every single one of those different verticals that we've spoken about. So it is both an art and a science to be able to figure out which one to go down. So industry is going to be the starting point for you.
You've got to know its strength. The strength is it's in solution, like clogged pipes. It works with that because clogged pipes are like that because that slime is just connecting all of the rubbish in the pipe and blocking it. All you need to do is release the pressure a little bit in the slime and it's gone. And our stuff can do that. So just need to activate that dispersion system. It'll open up and there goes their strength. The bacteria dissolve, the slime's gone and that'll unblock. So I know that's its strength. That's what we're going to aim for. It's going to help heaps of industries and then there might be some investment money, some money there to develop into other surfaces. Like it works. It doesn't work well on a surface on its own because what it does is it takes the top off, but it doesn't affect the bottom. So you need another molecule that goes with that as well.
Okay.
Things like that. So I know its strengths. I know what we need to do.
If I was going to frame that, it sounds like if we go back to the semiotics, it sounds like you need to develop a language of interventions. So you might have one language or one way of communicating to remove that top layer or make that accessible. But then it's like, how might we communicate with other elements? So how might we, cause it to detach, or how might we cause it to diffuse? And it's going to be sort of figuring out which ones of those, work for which context it's in, rather than a blanket, like, let's just use this one product for everything. It sounds like there's going to be different sort of, I guess, ecosystems of products that are going to be coming out based on. Okay.
Correct.
Interesting.
Yeah.
Cool. All right. Last question, I reckon. If you could run one experiment at a planetary scale, what would it be?
It'd be to get every hospital in the world to begin using natural ways to treat all of their sinks and their surfaces, instead of things they're currently using, because that would stop infections in hospitals by 50% at least, and antibiotic resistance increasing. I think I'd do something like that. It's going to make a difference in make a difference for the planet and also the people at once. Because you're not getting antibiotics going down the drain.
And reduce all-cause mortality at hospitals by so much.
You could do that. If I could do that, that'd be really, really huge, I think. I think that's the best thing to do.
Nice. All right. Well, is there anything else you'd like to share, or any parting words before we wrap it up and I let you escape back to the lab?
If you've got an idea out there as a scientist, or even not a scientist, if you've got an idea you think would work, might change something you want to do or build something you want to do, no matter how old you are, give it a shot. Got nothing to lose. You might find it's a stepping stone to something else. If you need some people around you, see if you can find them, or come to CoLabs. Come and chat with us all.
I like that.
Everyone's got ideas, from whether you're 3 or 4 year old up to 100. Everyone has ideas, and don't regret not doing them. You never know, you might change the world.
I like that. Yeah, and it feels really like doing that in an environment that is supportive is super useful and needed. So yeah, thanks for the little plug at the end. Appreciate it.
That's right. Thank
you.
you.
Awesome. Well, thanks again, and yeah, look forward to the next one.
Hope that was okay.
Yeah, perfect.
Thank you for tuning in to another episode. Thank you for listening all the way through. My gosh, I still can't believe people do this. It's a delight to be able to bring these conversations, talking about innovation that's happening at the edge of emergence to you. I hope you enjoy them. If you know anyone who is working on any form of innovation or impact-oriented initiative that is helping humanity transition towards a more circular, bio-based and regenerative way of being and doing, please put them in contact with us. We would love to find ways to support, help out, and explore potential symbiotic ways of relating. Thank you.